Hello, my dear students. How are you today? Let's start our English lesson. And today we will talk about one of the common diseases about influenza. Influenza is an acute infectious disease which occurs in epidemics and is caused by a virus. It is characterized by an abrupt onset and such manifestations as general intoxication and affection of the respiratory tract mucosa. Influenza takes the leading position in the human pathology. Influenza and other acute respiratory diseases constitute about 75% of all infectious diseases. The main thing is that, besides relatively mild cases of the disease, there are severe cases resulting in disability and sometimes death when children or old people contract the disease. If to say about the history of this disease, the first documented pandemic of influenza occurred in 1889. It's supposed to have begun in China and then spread to all the countries of the world in the following 1.5 and 2 years. Etiology. Influenza A viruses can infect a variety of different host species, an ability that is of great importance in determining their ability to cause pandemic infection in humans. Influenza virus type A was the first to be isolated in 1933 by intranasal inoculation of the ferret. In 1940, influenza virus type B was isolated. Clinical features. Clinically, influenza C causes an afebrile upper respiratory tract infection, usually confined to young children. Outbreaks are not recognized. Complications of influenza. Primary influenza pneumonia may occur, especially in young adults, during an outbreak and can be fatal after a very short illness of sometimes less than 1 day. A similar rapid illness can occur in the elderly. More commonly, a bacterial pneumonia caused by Staphylococcus aureus or Streptococcus pneumonia occurs late in the course of the illness, often after a period of improvement, resulting in a classical biphasic fever pattern. And now let's watch the video about influenza. Be very attentive, listen and watch carefully.
Influenza A is one of the most common life-threatening viruses in the United States, each year infecting approximately 10% of the U.S. population and causing about 20,000 deaths. Influenza is normally transmitted by droplets or aerosols from the sneeze or cough of a nearby infected person. When another individual inhales the droplets, the virus has a chance to invade the cells of a new host. Influenza A contains a genome of single-stranded RNA segments, enzymes required for viral replication inside a host cell, and a shell of matrix proteins. The virus also has a phospholipid envelope with several embedded proteins, including a molecule called hemagglutinin, abbreviated HA or H, which plays an essential role in viral cell entry. An influenza virus binds to host cell receptor proteins that contain polysaccharides terminating with sialic acid. A sialic acid that is attached to galactose provides a recognition site for the virus's hemagglutinin protein. The sialic acid can be connected at different positions on galactose. This example shows an alpha-2,3 linkage between the number 2 carbon of sialic acid and the number 3 carbon of galactose. The avian influenza strain H5N1 recognizes mainly this alpha-2,3-linked version of the carbohydrate, which is found in birds. In humans, such carbohydrates are found only deep within the lungs, which may explain why the H5N1 strain, though deadly, is rarely transmitted between humans. In contrast, the upper respiratory tract of humans contains mainly alpha-2,6-linked receptors, and it is to these receptors that human influenza A viruses bind. The hemagglutinin complex consists of a trimer of subunits. Each subunit includes a domain that passes through the viral envelope and a domain that binds to the sialic acid receptors on the host cell. For clarity, let's look at just one of these three subunits. For an influenza virus to become infective, hemagglutinin must be cleaved. An enzyme released from the epithelial lining of the human respiratory tract performs this function. The cleavage frees one end of a segment called a fusion peptide. The fusion peptide is hydrophobic and buried within the core of the hemagglutinin trimeric complex. The host cell takes up the virus by endocytosis. The endocytic vesicle then fuses with a lysosome, and its interior acidifies. The lowered pH induces a conformational change, shifting the receptor binding region back and triggering the fusion peptides forward to penetrate the vesicle membrane. A number of trimeric hemagglutinin molecules in the same region of the membrane mediate fusion between the viral and host membranes. The fusion process expels the contents of the virus into the host cytoplasm. The virus is ready to begin its replication cycle.
Treatment. During the epidemics, 10–25% of the adult population fall ill with influenza. 1–2% of them need hospitalization. Most patients are treated in polyclinics. During the outpatient reception or home visiting, it's always necessary to find out one or another patient needs to be hospitalized or can be treated at home. The severity of the patient's condition determined by the intoxication degree, complications, and the presence or acute conditions of the preceding diseases must be taken into account. A patient with such symptoms as high temperature, consciousness disorders, convulsion syndrome, repeated vomiting, meningeal symptoms, hemorrhagic syndrome, respiratory and cardiovascular insufficiency should be hospitalized. And now please answer to the following questions. How long do influenza symptoms last? What is the difference between influenza A and B? What are the first symptoms of flu B? And the last one, what flu is going around 2020? I hope that everything is clear for you about influenza. Now the lesson is over. Thank you for your attention. Goodbye.